Once I sit down,
all right, if you guys want to come in from the back there, we're getting started. So welcome, everyone. My name is Logan Walsh. I'm the Development Director here at GMGI. Thank you so much for coming. For those of you who found us, found this event online or in our newsletter, this is the third part of a three-part series discussing our research and how our research impacts various topics affecting our world. So it's been a great show from the community, and we're so excited to be able to offer these types of talks to everyone like yourselves. It really helps us to showcase what can happen here at GMGI. So I'll start with a real quick introduction to GMGI. So our mission is to address critical challenges facing our oceans, human health, and the environment through innovative scientific research and education. And we do this by following a triad. So the first part of the triad is the Research Institute, where you are now. So this is a research institute, very unique in the world. It's powered by genomics. And for those of you that went on a tour with Andrea, you saw firsthand some of our incredible capabilities here. And you hear more about some of the research happening here tonight. The second part of our triad is the Gloucester Biotechnology Academy. So we train local high school graduates to become entry-level lab technicians in the growing biotechnology sector here in the Commonwealth. It's a transformative program. It takes young people that perhaps are not on a path to career or a path to college and gives them a new step in life. It really does open the door to a field in biotech that would otherwise likely be closed to them. So it's a very exciting program. That's located at Blackburn Industrial Park. If anyone here tonight has not been there, connect with me afterwards. I'd love to show you our academy space. And then the third part of our triad, which kind of brings it all together, science community. We think Gloucester, we think Cape Ann is a tremendous place to do science. We want to attract more companies like GMGI or our neighbors Heffering or LifeMine upstairs to locate here in Gloucester. We have a trained workforce from our students. We have research that can prove that this kind of science can be done here. We want to continue to grow and catalyze the regional economy. So GMGI and you, help us, spread the word. Tell your friends that you came here. Tell your colleagues and family. Our network is what has helped us to grow to this point, and we are always welcoming more people to come here and see the type of impact that we're having. And support us. Every single dollar, and I'm not just saying that because I'm the head of development, truly does have an impact here. Everything you see here has been largely built by the individuals who are inspired by the work and impact that we can have, and everything from private philanthropy to foundations to support from local and state governments has been tremendous. So if you are inspired tonight, feel free to reach out to me about supporting GMGI. So I'm going to turn it over to Kate Castellana, who is our postdoc, to introduce our speakers tonight.
Hi everyone, my name is Kate. I'm a postdoctoral scientist here. My research focuses on building marine organisms as models to understand human diseases such as cancer and longevity, and I work very closely with Andrea on the sea urchin project, which you'll hear a little bit about today. And so I'm very excited to introduce our two speakers today. First is Dr. Marc Vidal, who is a professor of genetics at Harvard Medical School and the founding director of the Center for Cancer Systems Biology at Dana-Farber Cancer Institute, and also a co-founder of GMGI. He was originally trained as a bioengineer and geneticist, and he pioneered the concept of interactome network modeling, which means to look at the complex interactions of proteins in the cell. And in looking at the human interactome network, Dr. Vidal and his colleagues have been able to start unraveling fundamental relationships between cellular systems and human disease. Dr. Vidal was elected to associate member of the Royal Academy of Sciences, Letters, and Fine Arts, and the Royal Academy of Medicine, and has received several other prestigious awards, including the Abbott Research Award. His work has also been featured in several documentaries, interviews, and publications. And he's also included on the list of the world's most influential scientific minds in the biology and biochemistry category. And our second speaker is our very own Andrea Bodnar, who is the Donald G. Combs Science Director here. Her scientist lies at the intersection of marine biology and human health, and her experience in both academia and industry brings a really unique perspective and experience that has been really influential and important in establishing GMGI as a world-class research institute. Prior to GMGI, she was a senior scientist in the Molecular Discovery Lab at the Bermuda Institute of Ocean Sciences, where her research focused on using sea urchins as a model to understand cellular and molecular mechanisms underlying extreme longevity, aging, and cancer. And prior to that, Andrea held senior scientist positions in the oncology department of Roche, the Department of Cell Biology and Pharmacology at Huron Corporation, and the Bioprocessing Technology Institute at the National University of Singapore. Wow. So, two wonderful speakers tonight. I hope you enjoy, and let Marc take it away.
Thank you so much. It was nice hug for that. It's really embarrassing because when you go to a meeting and it's a bunch of graduate students that you've never met before, you go and they tell you these things and you go, okay, that's just read it. But in front of friends and colleagues, like, if my mom was here, I would even be more embarrassed. And since I'm talking about you guys, we have one elected official here today. That's Margaret Ferrante. She's been unbelievable. Should I go out? What? I mean, COVID is back. Okay, I'll do that. Can I turn? Yeah. Okay. All right. All the weird thing is, look at this. It's just like I'm always dressed the same way. Kind of boring. All right. Sorry. And one more thing before I start. Cancer is not easy to talk about, especially when you're with people who are affected. Okay, so what is, in my mind, the future of cancer research? You ask the question to 5 scientists or 50 scientists, you'll get 100 responses or something like that. So I'm just going to give you my biased view about this, and we can discuss it later. And then it will relate to, and my main job is to introduce Andrea Bodnar's talk. But let's start with a little bit of basic numbers. You've seen this before, but just to sort of have all of us in the room understanding this at the same level. Worldwide, cancers with an S, because really, as you know, those are many diseases, really, depending on the tissue and the organ that's affected, and the kind of cancer that's developing in that particular organ and tissue. But if you take all cancers together, you're about second in causes, number of deaths, cardiovascular diseases being still higher, but then everything else is below. So one of the things, since we talk about the future of cancer research, I've been in this field since 1984, and the question is always, with all this money being spent, both in the private sector and in the public sector, are we making any progress here? Okay, and when you go on the CDC websites or whatever, the serious source of information, it feels like the death rates are not really going down that much. Like, what are we doing? Why is it so hard? I just want to say one thing about that, and that is that when I was born in 1962, I'm going to turn 60 very soon, the life expectancy in the US was 70 years old, 70. And it's very close to 80 right now, if you take all people, male, man, woman, etc. So we have an aging population. That's one thing, which is not on this slide. What is on this slide is the age-specific cancer death rate. So if you separate people by age groups, you obviously see, and you do the right controls for the scientists in the room with a denominator of 100,000 populations. You see this amazing effect. So all of us, as we age, and it kind of makes sense because cancer is a huge perturbation mechanism that occurs in our bodies, and the older we get, the more likely we are to have accumulated what I'm going to be talking about in a second, all the events that lead to that, right? So, but if you then do an age-adjusted cancer death rates, I started at the Farber in 1999. That's when I started my lab. Actually, January '98, technically speaking, but I was just talking kids. That's funny. So 2 decades. Blue is total. This is man, and this is woman, by the way. We still have that huge difference. But you can see that in all 3 cases, we are doing better. Again, it's all controlled, like I said, age adjusted, etc. So we're making progress. And you've heard, I'm not going to cover that, we don't have time in 10 minutes. You've heard of immunotherapy, you've heard of targeted therapies, you've heard of combining them, you've heard of early detections, you've heard of better procedures to eliminate tumors, etc., etc. So all this together leads to this situation. I just wanted to give you one more piece of context here. This is all death in the United States relative to cancer and heart disease, and this is COVID in 2020. Okay, so you can take whatever you want out of that. I'll say that we talked about COVID a lot relative to how people are still affected by cancer, and even more in 2020 because of all the stuff like the delay in procedures, etc. Anyway, that's where we are. So why is it so hard? Why has it been kind of slow? Although we can see progress if we do the right controls and we do age-adjusted analysis. This is very simple, I apologize, but it's still very true. And that is that, all of us here have trillions of cells. We don't even know really how many cells there are in the human body. For the scientists, it's 10 to the 11 or 12 or whatever, something like that. And what happens then is from the very beginning of your life, from conception, cells have to divide. And if you want to get to trillions of cells, that means a lot of divisions, obviously, right? So things can go bad. We won't go through details. You can make mistakes while you replicate DNA or stuff like that. Graham knows very much about that. He's one of the leaders in the field of DNA damage and other things. But usually in our normal cells, when something goes bad, there's basically a mechanism that kills cells. They basically commit suicide, to summarize. Okay? But some of that goes away sometimes. Again, as we age, it gets worse, that the frequency of that actually goes up. And now what you have is a first mutation, a second mutation. A third mutation means a change in a gene, right? We know that, right? And that accumulates, and pretty soon you get uncontrolled growth, right? So you can see the beauty of our biology is associated with a huge disaster that we all have, above our head. We all are at risk in one way or the other, right? And the reason is that to make these amazing bodies that we all have, you have to have these repeated cell divisions, and things can go bad. I cannot say it in a better way than that. So to summarize and give you my sense of what I think the future of cancer research is, at least on my side of the fence, is we're going to go and use 3 words: genes, genomes, and interactomes. So with my Belgian accent, you might not understand, but it's interactome, meaning all interactions. Genome is all genes. So we're going to move from genes to genomes to interactomes, yes? In 2 or 3 minutes. And you're going to tell me when I reach 12, right? Okay. So you've heard of BRCA1, right? Angelina Jolie, remember the case? She had 83% chance of developing breast cancer or ovarian cancer during her lifetime. That's why she went to a double mastectomy, etc. Those are genes that, when mutated, can get to this problematic situation I showed you at the bottom of the previous slide, right? The fact is, we actually have a total of 20,000 genes in our genome, 20,000 parts. BRCA1 is one of them. And in the whole sort of context of developing cancer, BRCA1 would be just a little, would do a little bit of problematic, situation over here. But this is called the hallmarks of cancer. Again, Graham knows that very well. It's Bob Weinberg and Doug Hanahan, and Bob Weinberg is at MIT, right next to Graham's lab. But it takes a bunch of different things to get to a situation where a bunch of cells will become tumorigenic. At least 10 that we know of today. Let me give you an example. Most of the time, like I'm speaking right now, I have molecules in my body that prevent some cells to divide. They just can't divide, right? So what cancer cells do is they invade that. They figure out a way to not have to respond to that negative control, right? Avoid immune destruction. It turns out cancer is at some point so bad that the immune system that we have to protect ourselves against viruses and other things in the environment that are not us, that are not the self. The tumor is so far from the self that the immune system can start targeting it and to try to do something, killing it, etc. But again, cancers have learned by Darwinian evolution to avoid such immune destruction. As a matter of fact, that's the principle of immunotherapy. Is you go in and you change that, you basically sort of go around that capability of cancer, and now your immune system is much better at killing tumor cells in your body without touching the good cells. So you can see that it's more complicated than a BRCA1 thing that we might have heard about in the past. And so that leads to the second word is cancer genomes, and that is that at this point we know David Wall, one of the co-founders of GMGI, created this technology in the late 90s that allowed us, as a field, allowed the world to sequence by now tens of thousands of tumor genomes, maybe more, hundreds of thousands, I don't know what you guys would say. So we now have this big palette of all of the genes that are mutated. It's usually on the order of hundreds, depends on the cancer, depends on the cells, but say a couple of hundred genes are mutated in a way that is functionally relevant in every tumor. So that gives you a list of genes that have gone wrong by mutations. So the next step, the future of cancer research in my mind, is to try to connect all these genes together, to find how all these molecules are interacting with each other, and how all these interactions now drive a phenomenon that we all hate, which is moving from being a healthy cell to being a tumorigenic cell. Does that make sense? And we call that the interactome. So I just want to finish with that. I have a couple of minutes, or
You're at 11 minutes.
oh, that's good. That's why you didn't say anything. Okay, I could do this for 45 minutes. So this is a graph of work from my own lab in collaboration with a physicist called Laszlo Barbashi. And what we did here is we drew a network, as you can see, an interactome, where the little circles are different diseases you've heard or conditions. Obesity, diabetes, deafness, cardiomyopathy, and then leukemia, colon cancer. And you can see that they're connected by lines. And we put a line between two nodes here, two circles, if there's at least one gene that's involved in both diseases, in the pair of nodes that we're considering. So I hope you can see from this that when you hear BRCA1, that's one thing. It's a sort of a reductionist way of thinking about how you get to cancer. But that only happens in the context of a huge interactome in the cells, the result of which is shown here in terms of diseases. As a matter of fact, you can see also what's interesting here is you have a little neighborhood here where all the diseases, if you start reading them, they're all cancer related. Gastric, prostate cancer, anemia, thyroid carcinoma. Let me show you what that means. It's the same tools that we use to study social networks than we use to study interactome networks in cells. This is a high school social interactome, if you wish. Now the nodes are students, and the lines, or edges, as we call them, between them are friendships, self-declared friendships. This is actual data. This is real, right? You could also do this. Do you want to give me my computer? Thank you so much. I should have added this slide, and I thought of it too late. But this is someone's Twitter network, right? You know, so that person where I found this, I don't know where they are, is one picture, and all the other ones are those that are followers of that person or that that person follows, and then you go 1 or 2 steps, 2 degrees of separation from them. So you can see intuitively the complexity of this. Okay, a cell, and David Wald and I are co-authors on a paper that was just published, where a cell typically has, somewhere between 100,000 and 500,000 macromolecules called proteoforms. Now, for those of you who know the literature, so it's a huge problem to understand how these things interact with each other and how these interactions are modified to get to becoming tumorigenic. Okay, one more slide, because if you don't mention the New York Times, then people think that you're not doing the right thing. That paper was picked up by the New York Times in the late 2000s. You could go, it's gone now because it's been 14 years or whatever. But you could go live with your mouse. This is what the mouse did here. You could go with your mouse and click, and then all the genes in question would come and you would see their names and you'd see their potential functions. So it's like a map, if you want. These are interactome maps, as we call it. Now, introduction of Andrea. This sounds crazy, and it is. Okay. So, give you a sense, I just looked before I came, cancer genome in the Web of Science database that I use a lot to find papers has 20,000 papers in it. Cancer genome, with quotes. Cancer gene has 10,000 publications. Cancer interactome has 16. So you might say, well, that'd be.
How many are yours?
Won't go there. Ram is a scientist. Now it feels like a real conference. Now I'm not embarrassed anymore. So, and the reason is I really like to think outside the box, and so when the Broad Institute, for example, with gazillion millions of dollars, by the way, they do this very well. It's very important. Cancer genomes are extremely important. I tend to sort of think of the next thing then. I think Rick, for example, went there a few times in his career as well. So, you're sort of on the side of things. You're not like mainstream, but you have a sense that there's something coming on the horizon, that there's a new concept, that you're actually looking at future things. So when we came up here and started GMGI, and especially when Andrea joined us, actually we hired her mostly because of these possibilities and her amazing CV, and as you will see, her much better capability of giving a talk than mine. But the overall idea is, if you do this, we're so small. This is nice. Thank you for coming, by the way. We appreciate your time and all the help you've given us so far and the help you might give us in the future. But we're small. So how can you make a difference if you're very small relative to the world, basically, of science? Does that make sense? The Broad, UCSF, Stanford, MIT. You try to think outside the box. So what can a relatively small research organization do in the context of billions of dollars invested in cancer research? Think of the moonshot now, et cetera, et cetera. I think what you do is you sit down and you try to think in ways that no one else does, right? You can't be completely out there, because then you're wasting your time and other people's money, which maybe is what I've done all my life. But, my wife will tell you that for sure. So you think outside. So we realize, chemistry is going to be super important here. The major problem is, how do you find drugs, small molecules, that are specific, right? That target the problem without giving you the side effects that we all have heard about when people do chemo. For that to happen, you have to think outside the box. The chemists have, again, gazillion millions of dollars to do this. And we thought maybe the ocean can give us answers. And that's the first point that Andrea is going to cover, because she'll tell you why. But I think it makes total sense. There really aren't that many groups doing that, right? So it's thinking outside the box. And then when it gets to understanding the mechanisms of cancer, there's something, and I'll be done now. I know I'm being long. But remember I said at the very beginning, it's kind of almost surprising that it's the problem of making a human body and making it functioning for 90 years, let's say, hopefully, is such that it's actually surprising that someone gets to that point without having had cancer, actually. Honestly, I don't mean this in any bad way, right? I mean this as a scientist. And so there is that argument pushed to the extreme, which is there are species, like entire species, where you look at 100 individuals, 1,000 individuals, 10,000, no one gets cancer. How does that work? They exist, they're out there. And it turns out many of them live in the ocean. And so that's going to be the second part of Andrea's talk, which is to use that property of some species out there that don't get cancer. So it's like studying the thing the other way around, right? Thinking outside the box. You try to understand cancer by, giving it to mice, for example. That's good. But you can turn to a species that never gets cancer and say, why is that? How does that work? Anyway, so and then turn to ocean biology, learn from and take advantage of the complexity of the marine world is what we're about in this organization. So Andrea, sorry, go ahead.
Thank you very much, Marc. That was the perfect introduction, because I'm going to try and stay in the box. But I'm going to ask all of you to think outside of the box. Okay?
Oh, that's right.
Okay.
You also stood outside the box.
So for any of you who enjoy snorkeling or scuba, I think you agree with me that there's nothing quite so serene and peaceful and beautiful as exploring a new and diverse marine environment. This is a beautiful subtropical reef in Bermuda, and a picture taken by one of my former postdocs. But rest assured, as peaceful as this looks, there's a real battle going on down here. All of these organisms are competing for food, they're competing for space, they're trying very hard not to be eaten, and they're also trying to save room to grow. And they all have different strategies to do this. They're trying to defend themselves against infectious disease. This is a dog-eat-dog world out there. So if you're a fish or some kind of animal that can move, you kind of have the leg up on this survival game, because you can move away from danger, you can move to find food. If you're a clam or something that has a nice hard shell, you can protect yourself with that hard shell and just withdraw into your shell when you're threatened by a predator. But imagine for a moment that you're a sponge, or a soft coral, or a tunicate. You're attached to one spot, you can't move. You have no spines, you have no shell to protect yourself. So the way that you're gonna protect yourself is through chemical warfare. You're gonna make some really nasty toxic chemicals, noxious chemicals, such that it's gonna deter any predator from eating you. That just a nibble will basically send a message to that predator that, this could be your last meal if you keep on eating. If you wanna save room to grow, you're going to release chemicals that inhibit the growth of your neighbors so that you have space around you to expand. And so it turns out that these chemical signals, especially ones that inhibit the growth of your neighbors, turn out to be very effective at inhibiting the growth of cancer cells, which have uncontrolled growth. So it turns out these marine environments where you have a lot of different species all competing for limited resources are tremendous places to start looking for novel anti-cancer drugs. And in fact, there are 4 FDA-approved anti-cancer drugs on the market that come from marine organisms, and they all come from soft body, squishy marine organisms, 2 sponges, a tunicate, and the sea hare. Cytosar was the first FDA-approved drug in 1969. It comes from this little sponge, the Acrypta, which is found in the Caribbean. For those of you who remember your high school chemistry, if you can see that structure, it looks very much like a building block of DNA, but it has a very subtle difference in its structure, such that if it's incorporated into a growing DNA strand, no more building blocks can be added. So it stops DNA synthesis in its tracks. So it quickly stops actively dividing cells from dividing. Yondelis is the second marine-derived drug that was approved in 2005 for soft tissue sarcoma and ovarian cancer treatment. It's from this beautiful little tunicate, and it's an incredible structure. You don't have to be a chemist to see how exquisite that molecule is. It has a very, very unique mechanism of action, very unique structure. What it does is it binds to the DNA double helix in the minor groove and causes DNA to bend in a very unusual way, such that proteins can no longer interact with the DNA, and it ultimately leads to the death of actively dividing cells. Halogen is the third drug that was discovered from the seed, and it is used for treatment of prostate cancer, breast cancer, and lung cancer. It comes from the sponge, this kind of ominous black sponge, and again, a very, very exquisite chemistry. What it does is it binds to the cellular machinery that's necessary to pull cells apart at cell division and prevents them from physically dividing. Acedrus has a very similar mechanism of action. It prevents the cells from physically separating. It's an incredibly complex molecule and incredibly toxic. It actually can't be used by itself as a drug because it's too toxic. But how it's delivered is it's hooked up to an antibody that delivers it to the cancer cells, and it's used for the treatment of non-Hodgkin's lymphoma. So these are the success stories that have come out of the ocean, but there are many more in the pipeline. There are 28 marine-derived drugs currently in clinical trials, and most of those are for cancer treatment. And there are thousands of molecules in preclinical development. The ocean is turning out to be a rich source of new chemicals with really unique mechanisms of action that can be developed into anti-cancer drugs. So I just want to highlight 3 more because they have interesting stories. Apelidin is it's isolated from another sea squirt or tunicate. It's in clinical trials for treatment of multiple myeloma, which is a blood cancer, which really affects the bone marrow. But it's been in the news lately, and I don't know if any of you have heard about this drug. The way it works is it interferes with the machinery in the cell that makes proteins, and it turns out that it can interfere with the proteins that are made in viral replication. And so this drug actually is very, very effective at inhibiting the replication of SARS-CoV-2. And not only is it in clinical trials for multiple myeloma, it's currently in clinical trials for the treatment of COVID-19. It turns out that its potency is 30 times better than remdesivir, which is an accepted treatment for COVID-19. So again, we're seeing these really unique chemistries coming out of the sea. Briastatin is another really interesting molecule. It has a really interesting story with more twists and turns than a good mystery novel. It comes from this sea moss, this bryozoan, and that's probably familiar to anybody who has a boat or a dock. It's a fouling agent. It's a pest, considered a pest throughout the ocean. But in the 1960s, an extract of this bryozoan was found to inhibit the growth of cancer cells. So at that point, they invested millions and millions of dollars and decades of time to extract this beautiful molecule from the sea moss, to determine its mechanism of action, to try and figure out how to synthesize it in the lab, and to put it into clinical trials for cancer treatment. It's now been a part of more than 30 clinical trials, either by itself or in combination with other anti-cancer agents. However, after about 35 years of invested time and effort and money in developing this drug, in 2013 they stopped clinical trials because it wasn't showing significant efficacy over other available treatments. So it was really, it shows you how difficult it is, the drug discovery process. But one of the big hurdles of drug discovery from the sea actually is really well illustrated in this story. Because these molecules are present at very low quantities in these animals, in these organisms. And so you need to have a lot of starting material in order to extract enough of these compounds to actually use them in the lab. So in order to go into phase 1 clinical trials for briastatin, they needed about 18 grams of this drug to treat. In phase 1, it's a limited number of patients. So several hundred patients is mainly for looking for safety. So they needed 18 grams, and for those of you who don't think it in grams, this is 18 grams of salt, table salt. So it's maybe less than 10 teaspoons. So you didn't need very much, but in order to get this much briastatin from the sea moss, they had to start with 14, let me get this right, 14 tons of bryozoan. 28,000 pounds had to be extracted from the ocean to get enough to go into phase 1 clinical trials. So this has been a huge hurdle for drug discovery from the sea. You need so much starting material to extract these molecules that you can't do it without impacting the environment. So it was really difficult to do this in a sustainable way, and that's been one of the hurdles of developing some of these promising compounds. So that's why I want to highlight this molecule, because this is a real turning point in drug discovery from the sea. This is Salinasporin A, and there's a number of reasons why this is exciting. It's exciting because it doesn't come from a macrofauna or macroflora. It comes from a bacteria. It comes from a bacteria that was found in sediments in the Bahamas. And I want to point it out for a couple of reasons. It was discovered by Paul Jensen and Bill Fennicle. And Paul Jensen and Bill Fennicle are pioneers in the field of marine natural products discovery. Paul Jensen is a member of our scientific advisory board, and Bill Fennicle, a pioneer in the field, gave one of our science hour talks, which you can see on our website, about the history of drug discovery from the sea. It's also an exquisite molecule, but it has a really unique property in that it can cross the blood-brain barrier. And very few molecules can do this, and so it made it a very good candidate for treating brain cancer. And it's currently in phase 3 clinical trials for glioblastoma, very aggressive form of brain cancer. And so it's an incredible molecule with these incredible properties, but most importantly, it comes from a bacteria which can be grown in the lab. And so rather than having to go back to the ocean to source more and more material, you can, by a couple of approaches, you can make this drug in the lab in a much easier fashion. So the nice thing about bacteria is they have relatively small genomes and their genomes are circular. So this is the bacteria, the genome of this bacteria, Salmonespora tropica, from which Salmonesporin A was isolated from. And the other thing about bacteria genomes is they cluster their biosynthetic machinery together in the genome. So all the genes needed to make this molecule are in this little gene cluster inside that little red circle on the genome. So you have two options for making this in the lab. If this bacteria grows in the lab, you can scale it up by growing it in fermenters and isolate it. However, if you can't get that marine bacteria to grow in the lab, what you can do is take this little piece of, this piece of DNA, insert it into a type of bacteria that you can grow in the lab, and then grow that up in a fermenter and isolate the compound from there. So this really revolutionizes the way we can produce drugs from the sea in a sustainable way. And I want to take a minute here to point out that this technology, this bioprocessing and biomanufacturing technology, is really important in terms of producing new drugs and new compounds products. And this is something that our students in the Biotechnology Academy are being trained to do. This is thanks to a $900,000 workforce skills capital grant from tremendous support from Representative Ferrante and the state, which allowed us to build out a biomanufacturing teaching environment that provides the students at the Gloucester Biotechnology Academy with the skills to grow cells up in the lab on these large scales. And basically what happens here is the cells are growing in this environment where the cells themselves are the factory that are making the compound. And so it's a tremendous way to sustainably make the compounds. The other great thing about having drugs discovered in bacteria is that if you sequence the genome of bacteria, it's relatively small and easy to sequence. You can use computational methods to identify new biosynthetic gene pathways. So all of the little squares that are shown around the genome here are biosynthetic pathways, and you can use a computational approach to actually predict the structure that they'll make from the genes that are encoded in those biosynthetic pathways. So now you don't even have to extract the molecules anymore. You can extract the DNA, sequence the DNA, and use computational tools to predict what molecules are encoded in that DNA and make those molecules through a molecular biology approach. And so you have a couple of avenues now for making new molecules from the sea. So it turns out the ocean is a treasure trove of new drugs discovery. And if you think back to those original drugs that I showed you, the success stories that came from sponges and tunicates and sea hair, it turns out that the sponge, the tunicate, and the sea hair are not actually making the molecules of interest. It's a bacteria that lives in association with those organisms. And so really, it's not just sediments, but all of those other organisms. We can start to explore their microbiomes for all of these powerful drugs. So at GMGI, we're building a repository of marine microbes. We now have over 2,500 isolates of marine microbes that we can grow in the lab that we brought in from different marine environments that have the potential to make new molecules that could be developed into drugs. And we're also collecting genetic and genomic data that we can mine for new biosynthetic gene pathways. So as we begin to develop our own discovery processes and discovery pipelines at GMGI, we're also actively partnering with biotech companies and larger companies to try and leverage these resources for new discovery. The more hands we can get, the more hands on these compounds, on these cultures, and on this data, the more discoveries can be made. So we really want to partner and work with as many partners as possible so that we can discover and produce as many breakthroughs as we can. Okay, so if thinking about new drugs from the seed that are potent anti-cancer drugs isn't thinking outside the box enough, I'm going to ask you to think even further outside the box and ask yourself the question, what if we could prevent cancer altogether? And Marc introduced this a little bit, but nature appears to have solved this problem. And so what can we learn from her? Can we learn how she does this? And certainly in the marine environment, there are a few examples in which we see very few or no reported cases of cancer. And that's true for sea urchins, it's true for lobsters, and it's true for some marine mammals like the bowhead whale. And what's more remarkable about the fact that they don't get cancer is they all live very long lives, living between 50 to 200 years. And you'll remember that curve that Marc showed that cancer is basically an age-related phenomenon. You would expect animals that live 50, 100, 200 years to have a very high incidence of cancer after 200 years of living, and that's not what's seen. So these animals are doing something quite extraordinary, that if we can understand how they're protecting themselves from cancer through 200 years of living, can we then extrapolate some of that knowledge into preventative or therapeutic strategies for human cancers? Now, not only remarkable in that these animals live this long, they continue to grow throughout their lives, so cell division is continuing to happen. They regenerate tissues, so if you cut off the spines or the claws, they will grow them back. So they have the capacity for cell division throughout their lives, and yet they keep control of that for hundreds of years. Now, this isn't the case for all marine animals. There are some marine animals in which we see a very high incidence of cancer. Some species of bony fish are particularly prone to cancer. We know that beluga whales, about 27% of beluga whales that are found dead in the very polluted St. Lawrence estuary, 27% of them present with some type of cancer. And in marine bivalves, there are certain groups of bivalves in which there's incredibly high levels of cancer. Up to 90% of a population can have either some kind of leukemia, a blood cancer, or a gonadal cancer. Clams are a particularly interesting case of cancer because they are one of the very few species in the animal kingdom that can transmit cancer from animal to animal. So they have a contagious or an infectious cancer that spreads among the population. It happens very rarely in the animal kingdom. So there's a lot of interesting things we can learn from these marine animals. But my focus has been on these long-lived animals, and I've focused on sea urchins as a model because there's some particular challenges about working with big marine mammals and very, very aggressive lobsters. That you can only have one lobster per aquarium, or else it goes horribly wrong. But sea urchins are pretty passive, and you can put a lot of them in the aquarium. They've been a model for research for more than a century. There's hundreds of labs around the world use them as a research model, so there's lots of tools to study them, and so they've been the focus of my attention. Most people who study sea urchins study developmental biology, the beginning of life. However, I'm more interested at the end of the developmental spectrum in the aging part of life. So again, I'm a little bit outside the box in the developmental biology world as well. So we want to understand how these animals are protecting themselves from cancer. So there's a few things that we've done. We wanted to look at how their cells respond to DNA damaging agents. Now, as Marc mentioned, cancer is formed by mutations that occur in our genome in genes that control cell division such that we lose control of cell division. So we've taken cells from the sea urchin. We didn't know how to grow their cells in cultures, and we simply took the immune cells from these animals, and we treated them with a number of different things that cause damage to DNA: UV radiation, hydrogen peroxide, methylmethane sulfonate, and bleomycin. And what we noticed right off the bat is we had to give these cells very, very high doses of all of these treatments in order to even kill 50% of the cells. So the levels we were using are levels that would kill human cells in a petri dish, and yet these cells survive. So they have a very high tolerance for DNA damaging agents, which we don't really understand. We could measure DNA damage, and so we looked at DNA repair. They have very effective DNA repair. They share almost exactly the same DNA repair mechanisms as we do, and their DNA repair pathways occur as efficiently as they do in human cells. And when we induce DNA damage in the animal, we see an upregulation and activation of their immune response. So I think what's going on in these animals is they have some kind of first line of defense in which their cells are quite resistant to DNA damage, which it really behooves us to understand what that is, but we don't know at the moment. If they get damaged, they can repair it quite effectively, and if those damaged cells escape repair, the immune system comes in and removes them before we see the formation of cancer, or that's what we think is going on. But we want to be able to test this, and what we really need to do is develop the tools, the laboratory tools and the genomic tools to really test these hypotheses. So we've been spending some time over the last year or year and a half or so to sequence the genome of the long-lived red sea urchin. Once you sequence the genome and know all of its genes, you can start to understand what genes are playing a role in protecting this animal from cancer. So the sequence of this genome was done by Jennifer Polinsky, who's with us in the back room. She took a sample of the red sea urchin, extracted the DNA, and assembled its DNA into this beautiful scaffold. So the sea urchin genome has 773 million building blocks in its genome, and Jennifer managed to assemble these into 21 chromosomes. Each one of these little red boxes represents essentially a chromosome in this animal, and we can predict in these chromosomes there are about 24,000 genes present. So now we can dig into these genes and see what's going on with these animals. We have a list of all of the genes we know are protecting the human genome. So we have a special list of all those genes that basically safeguard the integrity of our genome, and on our list there's about 600 of them. When we look for the equivalent genes in the sea urchin, we find almost all of them. But what's interesting about that is in humans, for many of those genes that are guarding our genome, we have one copy of each of these genes, or many of these genes. The sea urchin has multiple copies of some of the key genes that are protecting the genome. So I just picked 6 examples here. These are called tumor suppressor genes, or genes that play a role in DNA repair. P53, retinoblastoma, P10, von Hippel-Lindau, CHEK1, and ERCC6. They play a role in keeping the brakes on cell division, they play a role in DNA repair, or they play a role in targeting damaged cells for cell death. So they have a variety of roles in protecting our cells. You can see in humans we have one copy of each of these genes. In sea urchins, they have one copy of P53, but they have multiple copies, 2 or more copies of the other tumor suppressors and DNA repair genes. So is this part of the secret of their success? Possibly, because it means many more genes need to be mutated before you are on the path to cancer. However, we need to be able to test this in the lab, and so again, we need better tools. So with funding from the National Science Foundation, we've been able to produce some conditions to grow sea urchin cells in the lab to provide a tool to look at how they respond to DNA damage and what's protecting them from cancer formation. So this is a project that I've been working on together with Kate in the lab. We've had some pretty good success. We've had their cells growing for more than a year in culture. It provides a tremendous model for us to now to treat them with DNA damaging agents, to look at the response, potentially build a nice interactome of all the things that are going on when these cells are stressed and what's protecting these cells from cancer formation. So I think all in all, we have a lot of different avenues to pursue to learn from the sea how to protect us from cancer and to discover new therapeutics for the treatment of cancer that are quite unique compared to what we're finding in the terrestrial world. So, thank you very much.
Thank you.
Any questions? Yeah, Rick?
What was the function of the gene that had 6 copies?
ERCC6 is a DNA repair gene, and actually Graham probably could tell you more about it than I can, but it's
probably part of nucleotide excision repair. Right?
That's right. It's part of nucleotide excision repair, and so part of the DNA repair pathway, especially in response to UV radiation.
Which means it sort of there's a system that scans looking for bulky lesions, and it sort of makes a little snip on the DNA on each side of the bulky lesion, takes it out, then you fill in a little gap, and you're okay. It's an accurate repair system.
And that some of those lesions are caused by UV radiation, which again, these animals, especially in their early life history, are exposed to UV radiation in the surface waters. So it makes sense that they would have really robust mechanisms to protect them from UV damage. I'm sorry.
Can you just repeat the question?
Oh, sure.
Yeah, so my question was, a lot of these chemicals that have been identified are kind of non-specific in terms of inhibiting DNA replication. How does the organism protect itself from the harmful effects of the chemical that it produces?
Yeah, so it doesn't actually. I think the difference is that, and Marc can probably answer this better than me, but cancer cells are growing much quicker than normal cells. Most of these molecules damage all of the cells, and in particular, quickly dividing cells are damaged. And that's why some of the principal side effects of chemotherapy affect cells in our body that divide rapidly, like hair follicles, the lining of our gut, our blood.
No, I was more interested in the organism that produces it.
Oh, okay.
The source,
okay.
the source of
Yeah.
how does the organism
How do they
protect itself?
cope with their own
That is actually,
guns?
that's an excellent question, and there's something very, very interesting as well that I want to answer that question, because I don't know, I'm not sure how they do it. It's thought it may tie in some cases to the ability to exclude the molecules from their cells. But there's actually some cases where there are organisms that will feed on the tunicates or the other organisms that have the toxic compounds, and they will not die, but they will sequester those molecules for their own defense. And so how that happens, I think nobody knows, but I think it's incredibly important to understand that.
And I have a quick related question. I mean, you talked about amplification of DNA repair genes.
Mm-hmm.
What about reflux transporters in terms of being able to overexpress things so you can push stuff out so that you can protect yourself from toxicity?
Yeah, and I think that's a problem for a lot of chemicals, but sorry, the question was, what about efflux pumps on the surface of cells that can pump out these chemicals, and is that a case for these as well? Is that what I understand? In fact, that is true for most of these chemicals, but what's very interesting about Yondelis is that one of the it causes some differences in transcription, and one of the things it does is it downregulates the expression of the pump, of the P-glycoprotein and the pump that pumps things out of the cell. So again, that's one of the mechanisms by which it is a very effective drug.
Andrea, you were saying that you are growing some of these different molecules or bacteria that produce those different molecules. What is your first key of assays that you're doing to see if anything will be, interesting and goes forward, or how do you screen? What is your first screen?
Yeah, so we have not developed our first drug screen yet. That's why we're partnering with biotech companies and maybe potentially someday partnering with Marc's lab where they already have established screens. So what we're doing is we have the repository, we can grow the organisms up, we can make extracts from the organisms or collect the supername from the growth block to be tested in various tests, but we don't have an ongoing screen at the
Or
moment.
like tissue culture, would you do this? That's what you would do your first step, I guess?
Historically, that's one of the ways that a lot of anti-cancer drugs were found through a collection of cell lines from the National Cancer Institute, the HL60 cell lines, where some are cancer cells and some are normal cells, and you look for which ones are the growth is inhibited. But there are many different screens targeting certain enzymes that play a role in cancer, and I don't know if you want to talk about your screen.
Yeah. So, and again, this is going to be a little bit technical, so for non-scientists, but I'm going to try to put it together. So, the epigenetic regulation of tumor cells is also extremely important. We didn't really touch on that today. And there's an enzyme called histone deacetylase, right? That is closely involved with that. So, and this is again, so it's a great question, Sandra. So the history of most HDAC inhibitors, by the way, for example, LifeMine is at least indirectly related to the discovery of romidepsin, which is Greg Vardine's company before, whatever, for those who know about this. So it's all, science is usually very well connected, as you know, Sandra. But the original search was for antibiotics. So for example, TSA, trichostatin A, or other classical tripoxin, other HDAC inhibitors in the 70s. It's actually a lot of Japanese groups were working on this. For some reason, it was something they would like to do. But then eventually, cell lines were starting to be used, and people started realizing it wasn't really an antibiotic, but it was an HDAC inhibitor, right? Problem with that is billions of dollars have been spent on developing HDAC inhibitors. They can work, but they're not super good. And they're also very restricted in the kind of tumors that you can solve, that you can do something about. So what we did, to answer your question, is I'm actually a yeast geneticist at heart. That's how I grew up in science. So I went back to my original love of yeast genetics, and I'm using yeast in my lab to screen for new HDAC inhibitors. And so the way it's done is the yeast cells die on a particular plate they're not doing well unless you inhibit an HDAC complex. And so we're looking for, among the 2,500 extracts that Andrea has, and also with Paul that you mentioned his work as well, they have about 10,000 reagents that we can use. We're looking for new molecules from the ocean that will act in yeast first, but then also in mammalian cells after that as new types of HDAC inhibitors. Yeah. So it's a first screen. Yeah, so what I love about that is I don't dive very much, but if I was a good diver, I could go down, get a few samples from the bottom, come back to my lab or here, put it on a Petri plate, and I'm asking the yeast if they can grow or not. And that's the start of it. So that's the link. Yeah.
I had another question for you. I mean, the aging effect in sea urchins, there's a lot of evidence in other model organisms that methionine metabolism is important, and if you restrict methionine, you can inhibit aging. Have you looked whether methionine metabolism is important in sea urchin as a protective mechanism?
Not specifically, but we have done some gene expression, age-related gene expression, and we do find that the mTOR pathway is downregulated with age, which is opposite to, would be beneficial and sort of ties into methionine metabolism as well. And that would have a beneficial outcome in terms of the lessening the degenerative impact of aging. So certainly there potentially could be a link there between the downregulation of mTOR, but we haven't looked at it biochemically yet. Yeah,
just trying to link your two talks. Just asking, sort of out of the blue, but I was curious whether the sea urchin had BRCA1 and 2. If so, did it have multiple copies?
It has one copy of each. So initially I thought there were multiple copies, but when I looked at them, it was a gene that was broken up into two, so it got stitched back together in our analysis. But
And
it's only one.
for the rest of you, what those two ultimately end control is the repair of what they call a double-stranded break, which is the worst thing that can happen to DNA with both strands break, and you somehow gotta get it together, and that's what those guys do, and if you do it accurately, you put it back together. They help with putting it back together.
Is there any regulation or agency that you work with in regard to the protection of the ocean? And, you know, if you come up with something that you say, oh yeah, this would be great, but your example was 14 tons, as environmentalists in the ocean, that's a boatload, right?
Mm-hmm.
So then you stop and say, even though it may produce this drug, is it sustainable, is it environmental for the other end of things? Mm-hmm. So is in the equation, does that ever kind of come up, or can we reproduce this in the lab so we can go forward? But if we can't reproduce it, then maybe we shouldn't go forward. Is there any consideration?
There are always permitting. You have to apply for permits to collect anything from the ocean, and we go through all the state channels for collecting things. But this is why the new techniques, genomic techniques for discovery are really groundbreaking in terms of new discovery, because you need so little material to start with that you can, once you take a tiny bit of a sponge, you can sequence the genome, and that's all you need to move forward for drug discovery. Or you can take a tiny bit and culture the microbes that are in there, and that's all you need. So it's becoming less of a problem, but there was always a lot of permitting issues and environmental assessments before any of this was done. When they were trying to scale up Bryozoan in order to collect enough to do the clinical studies, a number of companies were started that were making all kinds of like box kites and giant structures to put in the ocean to naturally foul so they could harvest it. So to try and do it more sustainably than taking it out of the environment. So in general, people are, there's always been an effort to try and preserve the marine environment, and many compounds have been terminated because the impact was too great to take that much compound out of the ocean. So now we can go back and use the new technologies and genomic approaches and reassess the molecules. Okay, should we wrap it up? So we'll wrap it up and have drinks and food and more discussion.
So and let's just give Marc and Andrea one last round of applause. I think this is the exact kind of research research and work that we're so excited about here. So we're incredibly happy to share with you tonight, bringing these two topics together and showing you how marine genomics, the institute here, located in Gloucester, can really never be anywhere else but here in Gloucester. GMGI is really trying to lead the pack in a number of inquisitive, incredibly valuable pathways to help the ocean, human health, and the environment. So thank you very much. And as Andrea said, please stay, enjoy more food and drink. Talk to some of our research scientists. And if anyone didn't get a tour, would like a tour, come find me and we'll make sure you get to the lab. Thank you, everyone.